Hello students, welcome to my channel Online Biology Tutorials. We are studying chapter number 5, the fundamental unit of life, and we have reached the topic of ribosomes. Now ribosomes term was already being heard when we were studying endoplasmic reticulum. Endoplasmic reticulum, to brief you here, it was of two types. It could be rough ER or rough endoplasmic reticulum, or it could be smooth endoplasmic reticulum. Basically, we discussed about ribosomes in the rough endoplasmic reticulum. If you haven't watched the video on endoplasmic reticulum, a link will be shared in the I button on the extreme right side. You can click on it to check that first. Now ribosomes, as I have talked initially also, that they are the protein factories. Now when I say protein factories, and a factory is a place where a thing is being prepared. So basically protein synthesis happens with the help of ribosomes. Where are they being found? They are found freely in the cytoplasm. This is very important. They can be found in the cytoplasm as free components, or they may found embedded. They are found at both the places, embedded on rough endoplasmic reticulum. So these are the two sites where they are being found, either in the cytoplasm or they may found embedded on the rough endoplasmic reticulum. Now, what is the actual structure of a ribosome? How does it look like? So ribosomes, they are generally being made up of a smaller unit and a larger unit. So we are talking about one ribosome, and it is made up of small subunit and large subunit. Right? Now how to show the small subunit? This is how a small subunit looks, and this is how a large subunit looks. Whenever the ribosome is not functional, what do you mean by not functional? When it is not synthesizing or producing protein. Okay, whenever it is not synthesizing or producing protein, the smaller subunit and the larger subunit, they are apart from each other, they are separated from each other. But whenever we are talking about a functional ribosome, a functional ribosome would be the one that has to form the protein, because that's its function, right? So in the functional ribosome, the smaller subunit will be united to the bigger one, and this is mandatory. This is necessary for protein synthesis, that larger subunit should be joined to the smaller one. Okay, smaller subunit. So here in this representation, this one, you can see that this is the functional ribosome, because smaller and larger subunits are joined together. Now whenever we talk about ribosomes, we need to give a numeric value, right? We need to give a numeric value. Let's talk about prokaryotes ribosomes and the ribosomes that are present in the eukaryotes. This will become clearer to you then. In prokaryotes, the ribosomes are 70S, whereas in the eukaryotes, the ribosomes are 80S. Right? Here you can see that I have written the numeric value 70 in the case of prokaryotes and 80 in the case of eukaryotes. But this value is also accompanying an alphabet in both the cases. Here S means Sived-Berg unit. Whenever a scientist discovers something, just to honor that scientist, we give that unit to a particular discovered thing. So Sivdberg, he gave a concept of sedimentation coefficient. Sedimentation, S can be known as sedimentation coefficient, or it could be known as Sivdberg unit. Just to honor the scientist, we have named it Sivdberg unit. Now sedimentation means the process of settling down. You might have seen the things which are greater in density, they settle more faster than the ones that are more lighter, right? So in prokaryotes, ribosomes have a different sedimentation coefficient, whereas in the eukaryotes it is different. So in prokaryotes the value is 70s, whereas in the eukaryotes it is 80s. Now we will also see how the value of smaller and larger subunit varies in the case of prokaryotes. In prokaryotes and eukaryotes, of course, the ribosome will definitely have a smaller subunit and a larger subunit. Whereas in the 80S, same condition will be followed. Smaller subunit and larger subunit. The value of smaller and larger subunit varies in prokaryotes and eukaryotes. Now see here, it is 30S in the case of smaller subunit of prokaryotes and 50S here in the larger subunit of the prokaryotes, whereas here it is 40S and 60S. The students who are aspiring for the Olympiad, for them it is very essential to learn these values. That means if you are going to draw the smaller subunit and the larger subunit, here also the smaller subunit and the larger subunit, the value will be differing. It is 30S here, 50S here, it is 40S here, and it is 60S here. I hope it is clear to you. Please don't apply this rule that 30 plus 50 is 80, but why ma'am has written 70S here? This rule is not applicable, okay? This is based upon the density, how fast they are going to settle down. Basing upon this, the value has been given. Similarly, here also 60 plus 40 concept has not been applied. Overall, the smaller and the larger subunit, they should assemble together to make the protein. Now the question for this video is, what does S stands for in our ribosome? The students who will be giving correct answer to this question in the comment section of this video, their names will be taken in the upcoming video. Stay tuned for more learning videos like this, and do share my videos with your family and friends for maximum benefit, because your tests are certainly near. Thank you.